Okay, question 12. Congenital lactase deficiency in humans is caused by a very low activity of the enzyme lactase, resulting in individuals being unable to digest lactose in milk. This is caused by a number of different mutations in the lactase gene. One of the mutations involved causes a frameshift mutation in the lactase gene. Name a gene mutation which causes a frameshift. It's a bit of a gift. It is either insertion or deletion. Describe the effect of this frameshift mutation on the lactase gene and on the structure of lactase. So this comes up quite a lot, this idea of you have to give the effect of the mutation and then say how that would impact on the protein. Okay, so the effect on the gene is that all codons downstream, or just after the frameshift, are changed. Now what that means on the lactase is you've got quite a profound effect on that, because basically all of the amino acids are changed after mutation. And that's probably going to mean that you end up with a completely non-functional protein, because, I mean, it's completely changed, everything downstream of where the impact was. So if it's early on in the gene, you might only have like 100 or so amino acids that are as they should be, and then everything else has totally changed. That means your protein won't fold properly. It's just not got the same shape, and shape is function, function is shape. You know, you cannot have one without the other. So everything is altered. So the effect of the structure is, and so all structure changes. So the whole thing completely goosed. Okay. Right, some mutations occur in the sequences which regulate the transcription of the lactase gene. Suggest why this may lead to more lactase enzyme being produced. Okay, so if you've got something which is regulating transcription, so that is deciding how much you produce in terms of the mRNA ready to go and get translated. Why could it lead to more? Okay, so if the mutations are regulating the transcription, the sequences are regulating, and the mutation then stops that working. So you have something which, again, this used to be in the course content, but if I put this in here, okay, if here's the lactase gene, and we have a regulatory sequence here, okay, and say that regulation sequence has to connect on a particular protein, okay, and that particular protein connects onto that one, and while it's connected on here, nothing happens with lactase. But if you have a mutation inside this bit here, inside this code, then this bit can't stick on anymore. So now that that's no longer there, this just constantly makes the lactase be produced. Okay, so what we're looking at is if you remove the regulation and you stop it stopping the lactase being produced, you get more. Okay, now this part is no longer in the course content, but just for interest's sake, because it does link a bit to still what's there. Mutations in the gene are common in Finland, more common in Finland than in other parts of Europe. This is thought to be due to a small number of individuals who settled in Finland many generations ago. State the term which describes this change in gene frequency when a small population of individuals break away from a larger population. Now the reason I'm saying I'll still mention it is because the only one you still need to know is a bottleneck effect, and this is a type of bottleneck. This is actually what's called the founder effect, where you have a very small number of individuals that starts a new population, and obviously what you have there is all of the population that comes from that are going to have a higher proportion of the alleles that were in that original founder population. Okay, but you don't need to know that, but you do need to know about bottlenecks. Okay, that's it.